Hi, everyone. Thank you so much for joining us as we welcome Dr. Jim Estes on behalf of the Department of Integrative Biology. Dr. Estes is a renowned marine community ecologist, well known for his work on the cascading impacts of high trophic level consumers on the communities and environments in which they live. Dr. Estes received his undergraduate degree in zoology from the University of Minnesota, his master's in zoology from Washington State University, and his PhD in biology with a minor in statistics from the University of Arizona.
So while Dr. Estes now resides as Distinguished Professor in Ecology and Evolutionary Biology at UC Santa Cruz, he spent a majority of his career with the U.S. Fish and Wildlife Service and the U.S. Geological Survey. So he's a really great example of someone who has conducted outstanding and impactful research regardless of the avenue that he is currently working in, whether it's government or academia. He's received numerous awards that reflect this and his contributions to both science and society, including the Pew Marine Conservation Fellowship, and most recently in honor of induction into the National Academy of Sciences in 2014. But despite this VIP status that he holds, he remains incredibly humble and dedicates a lot of time to engaging with graduate students, exemplified by both his interactions that we've had with him here today. He's given us a lot of great insight into working in both academia and government, but also by being the keynote speaker for the Biology Graduate Student Symposium next Saturday, which is graduate students only. He's the only faculty member invited. So, with that.
So today, Dr. Estes will be presenting his talk, Sea Otters and Kelp Forests: Questions, Approaches, and Perspectives. So let's please join me in giving a big round of applause and welcome to Dr. Jim Estes.
Wow, thank you all for coming. So you can all see me standing here, and one of the things that's obvious is that I'm closer to the end of my career than I'm at the beginning. And so what I'm going to do as a consequence is I'm giving a retrospection of sorts. It's going to be kind of an overview of what I see as the high points of the things that I've been involved with over the course of my life. So let me begin at the end of sorts. And so here's an illustration. It's actually taken from a paper that I think some of you will recognize. It was published by Jordi Bascomte and colleagues in PNAS about 10 years ago. And it is a characterization of a food web, doesn't matter which one it is. And the little circles around the perimeter of the oval represent species, and the arrows that go back and forth across those species represent interactions. So my view of nature is that it's those interactions that are really important to us in understanding what it is that we need to understand about ecology, and that is, what is it that determines the distribution and abundance of species. In any introductory course, most ecology texts, the most fundamental explanation of the goal of ecology is to do just that. So it's not all species interactions that dictate the distribution and abundance of species, but they're very important. And so these lines exemplify a couple of things. One is variation in interaction strength. Some interactions are relatively weak, others are quite strong. It represents directionality. The interactions between species oftentimes have what we consider to be a driver and a recipient. So one species is really important affecting the other, not necessarily in the other direction. So I've made a little list here of 3 things that I think are very important in this sort of conceptualization of the dynamics of nature. One is the immense structural complexity of the interactions. So there are just short of 100 species characterized by this particular cartoon. If you calculate all of the number of potential pathways across those arrows, it comes out to be something in the range of about 2.5 times 10 to the 26th. So the diversity of nature is in species interactions and the complexity of interactions, and that's because of all of the potential indirect interactions and the varying lengths of those potential interactions. So that may seem unbelievable to you, but if you do the math, that's the number that came out. At least that's the way it came out when I did the math. I've talked about interaction strengths being important. The third dimension that makes understanding nature particularly daunting is that there are functional relationships to these interactions. That is, it isn't an all or none, with or without thing, but they vary as a function of species abundance. And so understanding the functional patterns of those interactions is also important. So those are the challenges, and that's one reason why, in my opinion, it's almost hopeless to think about ever really understanding an entire community and all of its species and all of its diversity. That's never really the goal. It may be some of the goal in theoretical considerations, but it's almost never the goal in empirical studies, and it's not the goal in the empirical studies that I have partaken in, and that's what I'm going to talk about today. So here, really, for me, is the beginning. This is the way I looked at nature when I started working on sea otters and kelp forests in 1970. I envisioned something that I called a coastal ecosystem that was all the species and the physical entities that live in the coastal, or that exist in the coastal ecosystem, and that's somehow impacting otters. And that's what had this vague notion of what I was setting out to try to understand. And then, after about 6 months, very fortuitously, I met this person. This is Bob Payne, a picture of Bob at about the time that I met him. I think this picture was taken in '69. I met him in 1971. And we had a chat, which I've talked to some of you about. And he told me that he thought my view of what I was trying to do was really not very interesting, and that, in fact, why not think about both directions of the way in which these entities might interact with one another. That is, not just the effects of the ecosystem on the otters, but how the otters might affect the ecosystem. And in 1971, when he told me that, or suggested that, it absolutely did not resonate with me at all, because I had no real precedent for thinking that that sort of thing was important, and it just seemed almost outlandish. But what did resonate with me was the potential for looking at this sort of process, and that was because of the history of sea otters in the Aleutian Islands. I knew that they were very abundant on some islands, and on the island that I was working on at the time, which was Amchitka Island, they were abundant there. But there were other islands where they had been abundant but were no longer abundant. And they were no longer abundant simply because of the vagaries of history. They had been hunted out and they hadn't recolonized those islands. And so whether or not this model was interesting, there was an approach to actually exploring the possibility that seemed very powerful. And that was the perturbation of this system and looking at a system where otters had been abundant but where they'd been taken out. And that was very appealing to me. So here's just a characterization of what that's all about. The Pacific Maritime Fur Trade, which began in the mid-1700s, stripped otters out of their entire range in the North Pacific. It left a few remnant colonies. Some of those remnant colonies recovered in the subsequent 50 years between when they were protected in the early part of the 20th century and when I came on the scene in 1970. And this really was the perturbation that allowed me to have some look into that interaction web or food web dynamic, and with a very explicit purpose of trying to understand what effect might sea otters have on this system. So this just shows where the remnant colonies survived, what the historical range of the species was in blue. And I was working at Amchitka at the time, shown there, a red dot of one of the surviving remnant colonies. The green dots over in the east are places that otters were subsequently reintroduced in an effort to repatriate the species. But this history of near extinction and recovery and the fragmentation that resulted as a consequence of that really provided the window into virtually everything I know and that I've seen or that I've done as a consequence of my research program. So we went over to Shemya Island, where otters did not occur at the time and still don't. And I'd been working at Amchitka Island for about a year, and I knew what Amchitka Island looked like, so to speak. And so this is what I saw at Amchitka Island, and it took me literally a nanosecond to sort of get the story when I stuck my head in the water over at Shemya Island, because it was a fundamentally different looking place. There were lots of kelp at Amchitka, and there was virtually no kelp at Shemya. There were very few urchins at Amchitka. There were lots of urchins at Shemya. I knew that otters ate urchins. I suspected, although it wasn't well established at the time, that urchins ate kelp. And so this was the image of the dynamic, which is an example of what Payne subsequently called the trophic cascade, of what I thought was explaining the dynamics of that system, and I still think that's the case. And that is, when otters are abundant, they limited urchins. That in turn released kelp, allowed the kelp to become abundant at places where otters were abundant. And when otters were lost, the urchins became abundant and they ate all the kelp, and that was the explanation. So nothing really new about that. But here are the questions that arose as a consequence of that initial observation and that have defined much of my research program over the following almost 50 years. And that was, okay, we see this interaction, how general is it? Does it occur, were we cherry picking? Did we go to a place where it just happened to work? I didn't really know that. I didn't think so, but I wasn't sure. And so we were challenged on that question, and I was actually challenged very vigorously on that issue by Mike Foster, as some of you will know, way back. And I was really pissed at him at the time because I thought he was blowing smoke. But in fact, it really forced me to do some things that I think were useful. So we spent a bunch of time trying to resolve that question. The transitional dynamics of going from a system that is all kelp to one that's all urchins as otters numbers wax and wane became an interesting question. So I'll tell you a little bit about some of the work that we did there. The broader consequences of this interaction to other species and ecological processes. So it wasn't difficult for me to imagine that a system that had a lot of plants in it was going to have a lot of other things that were different about it compared with one that didn't have plants. And so that became almost a logical fait accompli, but something that wasn't very well documented. And so we spent a lot of time looking at that. There are numerous strong interactions that go on in this system, and it wasn't difficult for me to imagine, or at least daydream, that those strong interactions played out over long periods of time would have evolutionary consequences, selective consequences that would play out in the evolution of these species. And so some of the work that we've done has addressed that issue, and I'll tell you a little bit about that. And then the last part, which was a complete surprise to me, because when I started the system, I was really focused on the notion of a contained system that didn't interact with adjacent ecosystems, that it didn't interact with the open sea, didn't interact with the land. Came to find out that that was a fundamentally incorrect view of the way it worked. So the last 20 years, I've been grappling with this question of inter-ecosystem connectivity. So I'll explain all these later, but let me just go back to the first of these issues, which is generality. And so what do? The issue was, how general is this trophic cascade? So here's my approach. Simply look at places with and without otters, or at places before and after otters were either not there and there, or there and not there, and we have both opportunities, and to take a random sample, a representative sample of the coastal ecosystem, and characterize what it looked like under both circumstances, all right? And we've done this in 3 areas. We've done it in the Aleutian Archipelago in Russia, we've done it in Southeast Alaska, and we've done it in British Columbia. So here's the approach. Just took an island. I know this island either has a lot of otters or it doesn't have any. I'm not sure about this particular one. We simply overlaid a grid on the island. We took the grid intersections with shoreline as potential sample sites, selected a random suite of those points and sampled them, and argued from that random sample is going to be as close as we're ever going to get to a representative sample of what that island looked like, and thus a big place with or without otters. So we did that. And the data in the upper left panel shows the abundance of kelp on the vertical axis and the abundance of urchins on the horizontal axis. And the dark spots are data from places where otters were abundant, and the open symbols are places where otters were absent. And so there it is for the Aleutian Islands. You get a sense of the degree to which it's general versus not general. So we don't get a single point estimate. But what we tend to see very strongly is that when otters are abundant, urchins are rare, pretty much uniformly rare, and kelp is abundant but variable. And when otters are absent, kelp is uniformly rare, but urchins tend to be much more abundant but highly variable. And that it's a very sort of parabolic type of a functional relationship. Most of that state space is unoccupied, so keep that in mind because that's going to come up again, all right? Those are the data for the Aleutians. The panel on the upper right, data for Southeast Alaska. The panels in the bottom here are data from British Columbia. These are from, this is from work that one of my former graduate students, Jane Watson, did. And this one is particularly interesting, I think, because she gathered data from 3 different sites: Barkley Sound, Checklasset Bay, and Cayucot Sound. Barkley Sound was continuously occupied by a lot of otters over the 20 years of her study. Checklasset Bay was Barkley Sound had no otters, as it still doesn't. Checklasset Bay had lots of otters then and still does. And Cayucot Sound was recolonized by otters over the course of her study. And so you can see the data points from 1988, 1994, 2007, and it all fits this model. And what we additionally have in this particular data set is a view of the transition, the gray points in the panel to the lower right. So there is something going on here. I think the interaction is broadly generalizable in this sense, all right, and that is broadly generalizable geographically. And so, as far as I'm concerned, that's the end of the story on that dimension, all right, on that issue. One could quibble in California about this. We haven't looked at it in this way. So that remains an open question. But another issue that came up was the transitional dynamics. How do these systems transition from being kelp-dominated systems to urchin-dominated systems, or vice versa? Is it a linear transition that occurs as otters become more and more abundant, urchins become proportionally more rare, and kelp become proportionally more abundant? Or is it something different than that? And so here's just a characterization of a couple of ways that it might happen. The upper panel shows sort of the linear type of a function where as otters become more abundant, you get a gradual change in the structure of the community. And the one in the lower panel shows a different scenario where not much happens over much of that range, and then there's a fairly abrupt change. So we had a bunch of data from a whole bunch of places that again just show the abundance of kelp, the abundance of urchins, and these are all the different places that we sampled over 20 years, some of which have no otters, some of which have otters that seem to be at carrying capacity, and everything in between. So a lot of these are islands that maybe are halfway recovered. Others are just a little bit recovered. Others are almost entirely recovered. And what we see is the exact same pattern in this state space distribution. Exact same pattern. So that to me is virtually definitive evidence that this is not a linear relationship. Because if it was, we wouldn't see this. We'd see something very different. So this is just another characterization of those data. They're aggregated on a larger scale. And what this shows is sites that are urchin-dominated, sites that are kelp-dominated, sites that are urchin-dominated from all of these sites that we have gathered. And using some fairly simple statistical techniques, we devised a discriminant function relationship that would define whether the system was an urchin-dominated system or a kelp-dominated system. And then what we did was we plotted and fit a logistic regression to a functional relationship between ecosystem state here, either all urchins or all kelp, as a function of otter density, from none to around carrying capacity, which is about 16 animals per kilometer of coastline in the Aleutian Islands. This is all data, these are all data from the Aleutian Islands. And so the blue line is a forward moving trajectory. So that's the way the system looked as it was recovering, as otters were becoming more abundant. These are data from Attu Island that I gathered over a period of about 25 years. And as you can see, this system remains an urchin-dominated system until it gets way out, very high otter abundance, and then the shift occurs. Going the other direction, it's a very different functional relationship. All right? It stays a kelp-dominated system for a while, and then there's abrupt transition back in the other direction. So this system not only shows a nonlinearity, it shows what we call hysteresis, and that is differences in functional relationship depending upon directionality of this perturbation. So we wanted to understand why that was. Why is it that we get, one, nonlinear relationships in this transition between urchin-dominated and kelp-dominated states, phase states, and why is it different depending upon directionality? And you know, it's a hard hypothesis to test in time because this takes decades for these things to unfold. So I had a graduate student at the time, Brenda Konar, who's at the University of Alaska now, and Brenda really had a brilliant idea, and that was just simply a space for time approach to the problem. And she recognized that there were little patches of kelp around here and there, and that the borders between those kelp patches and the urchin barrens were models of how these systems might be imagined to change in time. And so we were able to actually do experimental work and obtain answers to the dynamics of this transition over the course of a year, whereas it would have taken decades otherwise to do it by looking in time. So here are just a bunch of data that show the abundance of urchins and the abundance of kelp and some other stuff spatially across a boundary between a kelp patch and an urchin barren. And there are a bunch of different sites where these data come from, and you can see that there's some variance associated with it. But it again, is a very abrupt transition in space. So places stay kelpy until you get right to the border, and then they transition very quickly. So our approach was to do experimental studies at that boundary to try to understand the dynamics of what maintains these systems and how they change. So here's my little cartoon of what we really did. So this is the way it is in nature. You have a lot of kelp and then a lot of urchins and a very sharp border. And all we did was a sort of a simple set of experiments where we either added urchins to or removed kelp from this side of the border, or removed urchins from or added kelp to this side of the border, and looked at what happened. And when we did that, this is what we found. This is just a sort of a qualitative overview of the results. So when the system is in the kelp state, which is the solid lines here, kelp actually has a negative effect on urchins. And that's simply because the waves are moving the kelp around, it's beating the urchins away. So it actually has a negative effect on the urchins when there's kelp there. You take kelp away, urchins actually move in. The urchins, on the other hand, have no effect on the kelp when it's in the kelp state. And the reason for that is that there's all of this production and detritus falling down to the seafloor, so the urchins don't have to move at all. They just sit there and eat, and so they're not destructive grazers. So what you have is an amensalism under that circumstance between the urchins and the kelp. When you're in the urchin-dominated state, it's quite different. The kelp actually has a very positive effect on the urchins, fuels them, puts them in better shape, makes their gonads bigger, all that sort of thing. Their growth rate goes up. And the urchins have a very negative effect on the kelp. So in that state, it's a classic agonistic predator-prey, consumer-prey interaction. So depending upon the state that the system is in, the dynamics of these species interactions are radically different, shifts from being amensalistic to consumer-prey, and somehow, and I haven't quite got my head around this yet, I think that really helps us understand hysteresis in the system. Okay. Moving on to the third module, broader ecosystem effects. What does it mean to have kelp in a system? What does it mean to have otters in a system beyond the urchins and beyond the kelp? So we've looked at a number of different things. Here again, it's just a characterization, cartoon characterization of the trophic cascade. Otters mean kelp. Kelp means two things, at least, production and habitat, probably means other things. So we've looked at a number of what I would call knock-on effects of having kelp in the system on other species and ecological processes. So I just want to give you a real quick overview of some of the high points, some of the things we've learned. The approach was exactly the same as the approach we used before, and that is simply going to places where otters are abundant, going to places where they weren't abundant, but in this case, instead of looking at the trophic cascade, we're looking at other dimensions to the food web to see how different species are or ecological processes are. Did it in all these same places, basically did the same thing. It's very simple in concept. So I'm just going to show you now a series of slides that show some of the measurements that we did. So the first one, work that I did in collaboration with Dave Duggins and Cy Simonstead many years ago, where we looked at the growth rate of filter feeders in otter systems versus otter-free systems versus otter-dominated systems. And what we see is that about a twofold increase in the growth rates of mussels that are outplanted into systems where otters are abundant compared with where they're absent. No big surprise, there's lots more carbon, lots more production in the water column when there's kelp there than when there's not kelp there. So I think we would have been amazed not to get this result. But at the time, people thought it was pretty surprising. I'm not sure why in retrospect. Here's another illustration from a similar kind of measurements. This time we're just measuring fish abundance. In this case, it's the dominant kelp forest fish in the Aleutian Islands. It's a hexagrammid rock greenling. And there's about an 8 to 10-fold increase in fish abundance in kelp forest compared to urchin barrens. That's an otter effect. This is actually the results of a master's thesis from one of your former students from way back, Dave Irons. And this was actually a stunning result, I think, because what it shows is the dietary composition of another consumer in the system. These are glaucous-winged gulls in otter-dominated systems in black. They are virtually entirely piscivorous. They eat very few invertebrates in those systems. Go to a place where otters are absent, they eat almost no fish and almost entirely invertebrates. So you're getting a big effect on the diet. Here's some data from another one of your former faculty members, the late Bob Anthony, a very close friend of mine. We worked together in the Aleutians on eagles and other things. And what this shows is a large body of data that Bob accumulated from surveying hundreds of eagle nests across the Aleutian Islands when otters were abundant, and then about 15 years later after they had collapsed in the system. And what it shows is the proportion of seabirds, fish, and mammals in the diet when otters are abundant versus when they're not abundant. And what you see is that they become much more strongly focused on seabirds as a dietary element under this circumstance. And this actually has a very interesting knock-on effect because seabirds are very wide-ranging, whereas the marine mammals, which are mostly otters that they're eating, and the fish are not. And those wide-ranging seabirds actually interface in many places with a lot of contaminated oceans. And so what we saw as a consequence of this was an increase in the contaminant levels in the eggshells of the eagles as a result of that. The last little piece I want to tell you about was more recent. This was work I did in collaboration with a number of people, including Chris Wilmers at Santa Cruz. This was Chris's idea. He came to me one day shortly after he came to Santa Cruz as a new assistant professor, and he said, do you think this trophic cascade could have any effect on atmospheric carbon or ocean acidification? This was about maybe 10 years ago, and I said, how could this be? It just seemed insane that this would be of any consequence. And he wasn't so convinced that that was true, and so we agreed to look at it, and we looked at it very much in just a back-of-the-envelope way. That is, we knew we had information on all of the things that we needed to actually make the calculation. So we knew how abundant kelp was in places with and without otters, and we knew how much habitat there was, and we knew everything else we needed to know. We knew how much carbon there was in kelp. We knew what the net primary production rate of kelps were. We knew how much carbon there was in the overlying atmosphere. Everybody knows that. You know, that's out there for everyone to look at. So it was a pretty easy thing to just balance the books, just to do kind of an economic as I said, a back-of-the-envelope calculation of how much of that atmospheric carbon might be drawn out of the atmosphere into the kelp forest, depending upon whether otters are there or not. And so when we did that calculation, turns out it's a lot. Turns out it's about 10% of the overlying carbon in the overlying atmosphere would be sucked into a kelp forest if otters were there, that would not be sucked in if they weren't there. And it turns out, if you value this carbon, which people had done, so carbon at that time, and it's still being done in the European futures market, was valued. And so we used those values and we calculated how much would that carbon be worth to people if they were to sell this carbon in a futures market. And the standing biomass over the area that we worked in, which was the Aleutian Islands to British Columbia, was about $200 to $400 million. That's significant because there's a big conflict over fisheries conflict with otters, shell fishery conflicts, and the dollar value of those shell fisheries pales compared to this. If we look at sequestration, not just storage, but not just the amount in the living biomass, but the amount that is sequestered through production, and depending upon what happens to that carbon, if it goes into the deep sea, and we don't know what happens to it, we don't know where the carbon goes. So it may immediately be remineralized and go back into carbon dioxide, or very quickly, or it may remain as organic carbon for a long time. If it goes into the deep sea, it will do that, or at least it will not be accessible to the atmosphere. And depending upon some of these scenarios, the value of that carbon, that additional production, could range anywhere from $6 to over $1 billion. So it's a big deal. I'm convinced. Chris convinced me that this is a big deal. And so otters are having some local effect. And that's not to say that this is globally important, but it does show that on a local scale, these species interactions actually can interface with the physical environment in various interesting ways. So the next part I want to talk about is the evolutionary dimensions to this. So I've talked about strong species interactions, and I don't think it's terribly difficult to argue that where you have a strong species interaction going on for a long period of time, especially where there's a negative effect, that there is going to be some sort of response, some sort of selective response in those species. So where we looked was at the plant-herbivore interface, because we knew there were strong interactions going on there that varied a lot depending upon whether otters were in the system or whether they were not in the system, and they were easy to study. So people had done a lot of work on plant-herbivore interactions in other marine systems, and before that, a lot in terrestrial systems. So we were drawn to that. This is work that I did in collaboration with Peter Steinberg, who was a grad student at Santa Cruz at the time and is now a professor at the University of New South Wales. So here's the idea. If we had a system with lots of otters, we would expect that they would limit the urchins and that there would be very little disturbance from the urchins to the kelp, and so the kelp would not be under any sort of strong selective pressure to accommodate to high levels of herbivory. Take the otters out of the system or go to a part of the world where they don't occur or never occurred, and we would expect to see strong interactions between the herbivores and the plants. And so is there any truth in that view, and what might we expect to see? Where should we go to do it? So we chose to compare the northeastern Pacific, where otters had been for at least 2 million years and maybe a little longer, or something like an otter had been in that North Pacific system, and the southwestern Pacific, where had never been anything like an otter as far as we could tell. But there were kelp forests or kelp forest-like assemblages in the southern hemisphere that were quite similar to these, different species. Tropics have been an effective barrier to the dispersal of species back and forth across the northern and southern Pacific, cold water Pacific. So we chose Australasia, or New Zealand and Australia, for a place to work on this for a variety of reasons that I won't go into, but that's fundamentally the reason, all right? So the first thing we did was we looked at the intensity of herbivory in these systems. So how intense is herbivory? How much plant material gets munched up by an herbivore in a system in the North Pacific where otters are present, in a system in the North Pacific where otters are absent, and in the southern hemisphere where they've never been? And is there any difference across those 3 different scenarios? And so here are the data. So here is here are data from Amchitka and Adak Island. The panel on the left. So I would do what Andy Blaustein does when I hear these. Is there no Blaustein story about the cell phone? Was that you? So, sorry, a little bit of a distraction. So the bars on the left are controls where we've caged the plant, and the bars on the right are these situations where the plant is out on the open seafloor and completely exposed to herbivores. So here's the difference between the control and the experimental treatments. In otter-dominated systems in the North Pacific, no difference. I mean, they're virtually identical. Nothing gets eaten. Over here, same data, kinds of data, from a system where otters are absent. Controls, hardly anything gets eaten. The ones exposed to urchins get chewed to nothing very quickly. 80% roughly of the biomass gone in 24 hours. And this is what really excited us, was the ones in between, which are all from New Zealand. These are all marine reserves in New Zealand, fell somewhere in between there. They didn't all get chewed to nothing, but it was very different than what we saw here. There was a significantly higher intensity of herbivory in that southern hemisphere system. So we argued from that in fact, the Southern Hemisphere is a place that has been subject to chronic herbivory over long periods of time, much more intense chronic herbivory than we see in the North Pacific. And so we then looked at the plants and asked the question, are they defending themselves against this increased herbivory, and if so, how? So Peter convinced me that we should look at phlorotannins. And I won't go into a long explanation of why, but suffice it to say that phlorotannins are in all brown algae, marine brown algae. They occur in all species, and they were known at the time to be deterrent against herbivores. They may do other things, but we knew that they were deterrent. There was good evidence, experimental evidence at the time. So we looked at phlorotannins. That's an example of one simple fluorotannin type molecule up in the upper left. There are lots of them. It's a chemically very complex group of compounds. Here's the frequency distribution of the proportion of total fluorotannins by dry weight across species in the Southern Hemisphere as opposed to the Northern Hemisphere. Staggering difference. So the average in the Southern Hemisphere is about 10% dry weight of those plants are made up of fluorotannins. In the North Pacific, the average, depending on whether you do it on a per biomass or per species basis, is somewhere between 1/10% and 0.5%. There's a huge difference in fluorotannin chemistry between the plants in the Southern Hemisphere and the Northern Hemisphere. And when Peter first went there, which was way back when, I don't remember when, but it was before we had email, and he sent me a fax, and the fax had this big fluorotannin, huge fluorotannin molecule, and it said, "I smell it." You know, so he knew that, and it has sort of an astringent type of an odor to it. So this was consistent with our view that this was a flora in the Southern Hemisphere that had much higher concentrations of defensive compounds than the Northern Hemisphere. So then we wanted to test whether or not there was any consequence of these different chemistries to the herbivores, because the herbivores living in the Southern Hemisphere were having to deal with a flora that was much better defended than the herbivores in the Northern Hemisphere. So how do we do that? Well, there were a lot of things we couldn't do because they were problematic. They just would not be good experiments. So we couldn't move plants around because it would be illegal. We couldn't move herbivores around because it would be illegal. Looking at plants per se is not a good way to do it because they vary morphologically and all kinds. So what we did was we simply extracted fluorotannins, purified them, and put them into an otherwise neutral grazing model. So that grazing model is just agar with dried up ulva that everybody loves to eat. And so if you take these little discs with agar and ulva and you give them to any herbivore, they scarf the stuff. So all we had to do was vary the fluorotannins in these agar discs, and we could do it in all kinds of cool ways. We could vary the concentrations, and we could use fluorotannins from the Northern Hemisphere and fluorotannins from the Southern Hemisphere. And so we did that. And here is an overview of the results. Let me just explain this graph to you a little bit. So on the left, or to the left of the red vertical line, are herbivores from the North Pacific. Across the top, there's several of them. To the right are some herbivores from New Zealand. Above the horizontal red, I guess that's red line, are data from New Zealand derived fluorotannins, and below that line are North Pacific derived fluorotannins. Concentrations are all the same. And so what you see is there's no difference in the reaction of the herbivores to source of fluorotannin, but there's a huge difference in the herbivore's response depending upon whether they're herbivores from the Northern Hemisphere or the Southern Hemisphere. And I should have said that these stars represent a significant deterrent effect. The NS represents no significant deterrent effect. And so you can just sort of squint at that and see that there's a lot less deterrence in the Southern Hemisphere than there is in the Northern Hemisphere. So we very boldly imagined that this was what's going on. That in the Southern Hemisphere, you have effectively a two trophic level system, or at least an even number of trophic levels. And as a consequence, the intensity of herbivory is very strong. There's been strong selection for defense in the plants and strong selection for resistance by the herbivores. So there's been in our view, a strong coevolution for defense and resistance in that system. And adding that third trophic level to the North Pacific appears to have broken that coevolution of defense and resistance. And that's, we think, why we see what we do. A few spin-offs of this. So if that's true, and certainly the chemistry is true, I mean, that's pretty simple. And whether the mechanism is true or not still is certainly an open question, arguable. And I don't want to convey the notion that I'm absolutely convinced that this is a true story. It just makes sense to me, right? So if it is true, then we might expect also some spin-off effects of these different floras on other stuff that's living there and other patterns and processes the system. So one of the things we did is we essentially repeated the sampling approaches that we used in the North Pacific. And remember, these are distributions of data that have essentially are unoccupied state space. That is, all the data occur along the axes for the reasons that I explained earlier. So here's just a reminder of the different panels on the left that I showed earlier. Same data from New Zealand. And it's a very different sort of association of herbivores and plants. Herbivores and plants do not live together in abundance in the North Pacific. Herbivores and plants commonly live together in abundance in the Southern Hemisphere. So I've been thinking subsequently a lot about some of the other species that were kelp eaters. One of them is this thing, the Steller sea cow. I've always wondered if it might not be that the reason the sea cow radiated into the North Pacific but nowhere else from its tropical ancestors was because it was such a great meal there compared with other places where it was nasty. Maybe, maybe not. But we just recently did some analyses. This is some work I did with Dan Doak, very valued collaborator. We work together, and I love working with him for a whole bunch of reasons. And what Dan and I did was we asked the question, could it be that the extinction of the sea cows was not simply a consequence of these animals being hunted, but a consequence of sort of a co-extinction with the loss of sea otters from the North Pacific? And so what we did was we went back to two data sets that we have. And one of them was a data set from the Aleutian Islands, where we were actually able to watch otters collapse and watch the kelp forest change. And so that's shown in the upper panel. And then I had access to a bunch of data from the Commander Islands, where sea cows had survived in large numbers. So when the Bering expedition crashed on Bering Island and overwintered there were over 1,000 sea cows, perhaps as many as 1,500 that were living on Bering Island. And by 1768, they were extinct. By 1753, otters, which were very abundant on Bering Island when the Bering expedition discovered the island, were extinct. That's because of the beginning of the fur trade. They wiped them out in less than 10 years. They were gone by then. So we imagined that the response of the kelp forests under these two different scenarios was very similar in terms of the time series of change that we expect in the system. So we asked, would that account for the loss of the sea cows in the Commander Islands? And of course, we have no data on how sea cows respond to kelp, but we have a lot of data on how dugongs respond to seagrass and how dugongs respond to the loss of seagrass from hurricanes and things, or typhoons and things of that sort. So we used those demographic data to parameterize a sea cow model, ran the model, and lo and behold, it turns out that depending on how you vary the parameters, doesn't matter a whole lot, by 1768, we would predict, using those dugong data, that the sea cow population would have gone extinct in the Commanders. Another thing that we've looked at is body size in abalones. So here are just a couple of different species of abalones, a little one, Haliotis varia, and the biggest of all, red abalone, Haliotis rufescens. So there's a lot of variation in maximum body size in abalones. And we have gone around, this is work I've done in collaboration with Dave Limburg at UC Berkeley, who's a malacologist and very interested in sort of the paleo perspective. And we've looked at the data on maximum shell length in different abalone faunas around the world. And lo and behold, The two faunas that have the very largest abalones are the North Pacific faunas, the Japan fauna and the Northeast Pacific fauna. And we know from experimental work that we've done that fluorotannins have a very strong inhibitory effect on growth of abalones. So it very well may be that both the diversity and size of these abalones are a spin-off consequence. Again, there's a lot of arm waving here. Okay, the last little piece of my talk, and then I'll stop, has to do with the issue of inter-system connectivity. So I started thinking about this a while back when we were looking at time series data on otter abundance from the Aleutian Islands. And by about the mid-1990s, it became clear that something was very wrong out there, that numbers were going down. So these are just fits of data to different time series from different islands. The one that I'm sorry I don't have in here, Clam Lagoon, data from Clam Lagoon on Adak, which had a bunch of otters in it, and that one went straight across the top. It didn't decline at all. So we used these data and made a number of inferences and came to the conclusion that the most likely explanation for this collapse of the sea otters in the Aleutian Islands was because of predation by killer whales. And I don't have time to go into all the details. It's published. The fundamental evidence is simply this. All of a sudden we started seeing these things in large numbers, whereas we'd never seen them before, that is killer whales. They started eating otters. We'd never seen them eat an otter before. They started eating otters, and then the otters started disappearing. Clam Lagoon, which was a place that the killer whales couldn't get to, didn't decline at all. And then we did a whole bunch of modeling work to try to come to grips with the question of what would we expect to see based on all the data that we had in terms of the number of attacks of killer whales on otters if killer whales killed every otter that was required to drive the population downward at the rate that it was observed to go down. And when we did that and we back calculated and looked at what we would expect to see from that tens of thousands of mortality events that would have been necessary to drive those numbers downward, we predicted that we would see 5, and we saw 6. And so that doesn't mean that that's what happened, but at least it means that the fact that we only saw 6 is not surprising, given that this hypothesis is correct. So a bunch of other stuff happened that wasn't so surprising. That through time, otter populations declined, shown in the top graph there, just a recharacterization of the data I've already shown you. The abundance of urchins went up with the loss of the otters, no big surprise, that's what we had seen for decades. The intensity of grazing went from almost nothing to very high, as you can see. The abundance of kelp as a consequence went from very high to low. And so what we think happened in this system was simply that there was a shift in this trophic cascade from a 3 trophic level system when otters were at the top of the food web to a 4 trophic level system when killer whales were at the top of the food web, and everything went to pot as a consequence of that, or changed. So why did that happen? This is the most controversial part of anything I've ever done. And we came to the conclusion that it was a consequence of post-World War II industrial whaling that drove the whole thing, or at least in significant part. That's a hypothesis. That's not a, it's nothing more than a hypothesis, all right? But here are the whaling data. So those little red dots that you can't see, but you can kind of see how many they are and where they are in these panels, show the number of great whales that were taken out of the North Pacific Ocean following the end of World War II. And why is World War II important? Because there was no whaling, there was not much industrial whaling in the North Pacific prior to World War II. At the end of World War II, both Japan and Russia were on their knees economically, and so General MacArthur, who was in charge of of managing the recovery of these countries, encouraged them both to turn their wartime maritime technology to fishing and whaling. And they did that, and in the course of about 20 years, they basically stripped all the whales out of the North Pacific. And so you can see how many were taken in the 8 years after World War II, in the following 8 years to the lower upper right, in the next 8 years, and then at the end, just before the International Whaling Convention and the end of industrial whaling, there was almost nothing left up there. And so this is what we imagined happened. These are data from the Aleutian Islands. This shows the number of whales that were harvested within 200 nautical miles of the Aleutian Islands starting in 1950. That's when about the first post-World War II whaling occurred in that area. Went up, down to nothing. And that's because they were all stripped out of there. And then what we see is a collapse of harbor seals. We see a collapse of Steller sea lions. And then we see the collapse of sea otters that I told you about. And so what we have proposed is that the loss of these whales has driven killer whales, which were not exploited, to essentially force them to broaden their diets. And by broadening their diets, they started feeding on species that were unsustainable because they were very abundant in the system. So we've done a lot of modeling work to ask the question, is that reasonable? Is it even possible that it didn't happen? And in both cases, the answer is yes and no. So this is sort of my grand view of the food web. This is going back to Darwin's metaphor of a tangled bank. That's what we're looking at. And a teeny little piece of an ecosystem, just those little few pieces that I've looked at. So I've drawn in the middle here sort of the fundamental part of it, at least from my worldview, which is the otter, urchin, kelp, trophic cascade, and how that's linked into a whole bunch of other stuff. So the solid lines represent things that are pretty sure are going on. The gray lines are bottom-up forcing effects that are consequences of this. The dark lines are top-down forcing effects, and the dashed lines are things that are more speculative. And you can look at that for a while, but I've told you about all of it. And so if you're really interested in this and want to learn more about it and sort of read more about the details and sort of the behind the scenes part of it and the personal part of it, I have a book that's coming out, and it'll be out in about 6 weeks or 2 months, and this is it. And so I'm sorry for the shameless advertisement of my book, but you can read that. And of course, many people worked with me over the years, had lots of support from various agencies and organizations, and that's it. Thank you.
Got about 5 minutes for a few questions.
I was wondering if the result of your evolutionary path can explain that hysteresis that you have shown to us before, going from a sea urchin to an herbivory-dominated system. So in the sense that when there are a lot of kelps, and presumably less herbivory, one would expect a high genetic diversity, because there is less gradient, and that would confer resilience to the species. And then on the other end, when there are a sea urchin dominated system with very few kelps, lots of herbivory, little genetic diversity, and therefore less of resilience, to build up.
So the question was, I'm repeating the question for the recording. So the question was, would there be a relationship between genetic diversity and resilience as a consequence of these very different effective population sizes that would have resulted from urchins and kelp in otter systems with and without otters? And I have no idea. I just don't know. We don't have any data on genetics. We haven't looked at that, and I just don't know. I don't know. So often over the course of my life, I have said when someone's asked me a question like that, it can't be, and then I've been proven wrong. So maybe. I just don't know. Sorry. Yes.
If the recovery of whales caused the decline of sea otters in the latter half of the 20th century,
It would be the collapse of whales.
Yeah, right. Well, what happened then to the what was the trophic dynamics like in the 1700s when there were lots of whales and lots of sea otters and lots of dugongs?
Well, I mean, it all makes sense to me, because if there were a lot of whales at that time, which we think there were, then the killer whales were feeding exclusively on the great whales. They weren't feeding on pinnipeds or sea otters, and so they should have left them alone, and I would have expected them to be abundant in the system, and they appear to have been. So that's the way I imagine it was. I'm just imagining it, though, because we don't really know. We know they were abundant in the mid-1700s because the fur trade records show us that. We don't have any data on what the kelp forest in the Commander Islands looked like during the 1700s. I'm making that inference based on the similarity of species and what we have seen in the Aleutians with changes in otter abundance. So I would expect, based on a world with lots of whales, and there's reason to believe that whales had been abundant in that system for a long time, for a very long time, that they would have essentially buffered this collapse that we had seen over that period of time. Jane.
So Jim, are you predicting the reappearance then of sea otters in that system as whales begin to recover?
So the question was, and it's a great question, would one predict that this system will recover as whales recover? And they are recovering. So they are recovering. Most whale stocks are recovering or are almost completely recovered in some cases. So gray whales are thought to have been largely recovered. Humpbacks are making a great resurgence. Blue whale numbers in the North Pacific are increasing. Sperm whales have recovered. They were depleted. They are recovering. Most whales in most oceans are recovering with a few exceptions. And the answer, and so that's a very appealing scenario that we would expect it to recover, but my guess is that it probably won't. And the reason is that I think there is so much complexity to these dynamics, and I expect there's a lot of things that have happened, behavioral changes and other things. And so if it does recover, I don't think it will recover very quickly and simply. And of course, that's total speculation. But when we first came to this idea, I thought, well, this is testable. All we have to do is wait and see. And now I'm not so sure that there's really logic to that argument. So it may be, and it may not be, but it wouldn't surprise me if it didn't.
Maybe you need to go into orca training.
A lot of orcas in the North Pacific. So the population in that Aleutian Islands, Southern Bering Sea, North Pacific area is about 5,000 animals. And about 10% of those are mammal eaters. So there are about 500 orcas that are marine mammal feeders. And they're eating a lot of biomass. So it's actually remarkable that anything's alive up there, given that. When you do the calculations, I mean, the mortality rates that you would predict from 500 orcas would have driven those otters to extinction in about 3 days, maybe a little longer, but not much. So I don't think all of those orcas are feeding on otters. I think probably just a very few of them pick that up. So I think there's a lot of complexity at that level that we don't understand, that sort of subspecies or below the species level variation and behavior, diet, that sort of thing. Please.
I found the lack of a gradual transition between the urchin and kelp-dominated ecosystems to be really striking, and I was wondering if in your observations you found some sort of third-party organism or abiotic influence that was accounting for that lack of gradual transition, or that helped the kelp overcome the predatory influence of the urchins.
So the question had to do with whether there were some additional complexities or maybe a third species or something of that sort that was involved in this rapid phase shift between the urchin-dominated and kelp-dominated states. And no other organism, not that I've seen or know of, these phase shifts actually have been seen in a lot of other parts of the world, not just where we've worked. So it's a fairly typical characteristic of a lot of different kelp forests. But it appears to be especially pronounced in the North Pacific and in the North Atlantic, where the species are very similar. And their histories are actually very similar. I don't know. Sorry. I just don't, I told you what I know about it based on the experiments that we did. They were fairly complicated. I don't think those really explain why we have a phase shift, but I think they help us understand a little bit of the complexity that depending upon the directionality, you have a very different sort of species interaction. Now, one thing I can tell you is that urchin behavior is very important in that phase shift. So when kelp is rare in the system, urchins are mobile. When urchins are mobile, they eat everything. They eat all the baby kelps that are settling, and they eat all the adult kelps. They grow up, they crawl up on them, and they eat them. They eat the holdfasts away, and that's what creates the barrens. And once the kelps have recovered in the system and you start getting detritus falling out from that high production overlying canopy, then the urchins just stop moving. And I think it's that behavioral switch is very sharp from movement to no movement. And that probably is the main explanation for the abruptness of that relationship. That's just my gut feeling. Yeah. Eric, you had a question, please.
In terms of the whales, are some of those whale species competing with humans for food? And if they are those populations ever likely to reach levels they were historically before we started taking so many things out at sea?
Yeah. Well, it's a good question, and there have been a lot of people that have worried and wondered about that. People, various folks, including people in my lab, have tried to calculate how much of the net primary production, say from the North Pacific, went into whales when populations of whales were at their historical high numbers. And it actually turns out to be a lot. It's like about 65–70% of the total net primary production of the North Pacific would have gone through the whale fauna, given how abundant we think they were before they were exploited. So that leads to certainly a possibility that human competition for those resources could have an effect on the recovery of whales. I don't think there's really much evidence that that's happening. Whales are recovering pretty rapidly. In some places they aren't, but the places that they aren't and under the scenarios where they aren't recovering, it doesn't make sense that it's a food problem. So one of the stocks of whales that's not recovering are Southern Ocean blue whales, the far Southern Ocean blue whales that live in association with the Antarctic ice. And they're not recovering, and nobody knows why. There's a ton of krill down there, and they feed on krill. They're exclusively krill feeders. So that's a mystery. But I don't think food makes much sense in the case of blue whales. So I don't know. I mean, it's hard to imagine that all of the things that we are doing to the ocean through exploitation isn't having some effect on whales, but how that might play out in the degree to which they recover, the rate of their recovery is I think, very speculative at this point. Do you have a question?
Yeah, you talked about urchins responding behaviorally to kelp abundance, but do you think they respond behaviorally to otters, like actually sensing the otters being around or sensing conspecifics being eaten or anything like that?
Yeah, they do. Didn't have time to talk about that. Jane Watson, my student, did some beautiful experiments. She noticed, she was actually sitting right on a spot when it was invaded by otters. This was someplace in British Columbia, and so she was working there and was diving when the first otters came in. And so the first thing she saw were these huge red urchin tests kind of floating down to the bottom once an otter had trashed it and dropped the test to the bottom. And what she saw was that when those tests hit the bottom, all the rest of the urchins split. And they created these big kelp halos. And so the recovery was this hugely fragmented, sort of heterogeneous patch recovery that occurred in large part because of that.
So unfortunately, that's all the time we have.